Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing with you the October-November paper 5 variant 4 and let's get on with this. So the paper here deals with the question where they have given you a beetroot, edible fruit, edible vegetable, sorry, beetroot with the outer skin and all. We know it's kind of, if you just slice through it, it gives out all the red pigment and it's having leaves as well. Then there's a main taproot. Okay. The student determined the concentration of sucrose solution that has the same water potential as the cells of the beetroot. This is what the student had to do. The student used distilled water, the stock solution of 2.0 mole per dm cube sucrose to prepare a range of sucrose solution. Each sucrose solution was prepared using a proportional dilution and a final volume of 30 cm cube. Complete the table 1.1 to show the concentrations that were made. So this table is given to us that, okay, so this is what the student is doing. The initial volume, the final concept, the initial volume the final volume of all the solutions have to be 20. So volume of 20, and then how this volume of this thing is being used. Okay, so let's sorry, the volume have to be the volume have to be 30. My bad. The volume 30 needs to be made here. So how is this done? You have the final concentration given to you. So these two standards are already given to you that the student is using these values to make the thing. Let me just create a different color here. Okay. So initially, this is given to you that this is the initial thing when you are using the final concentration of sucrose solution and you are using it as the whole, adding no distilled water to it, the volume stays 30. So volume of how all the entire thing is the sucrose solution which is being used with no added water. Then eventually, if it's the final concentration of sucrose is none, there's nothing there, and all distilled water there, and how much of sucrose solution has been used up? Zero. None of it has been used up. So the entire 30 cm cube will be of water. Here, the 30 cm cube will be of sucrose. So how do you make up the remaining dilutions? The final concentration is 2 and but this is 0. So what will we do? We'll just use the proportional dilution formula of C1, V1, C2, V2 and devise some own the things here. For example, I can dilute it to 2.0, from 2.2 to 1.5, I can make a 1.0 solution, I can make 0.8, and I can make 0.5. So using this standard solution, I'm making all the solutions by proportional dilution, using this as a stock solution, adding appropriate volume of distilled water and the volume of sucrose solution to get the final concentration which I desire. How C1 V1 C2 V2 works? C1 V1 C1 V1 are the stock concentration and the stock volume, and C2 V2 are the concentration that we want to achieve and the volume that we need to use for this particular for making this particular dilution. How much water will be added? Water will be added by 30 minus V1. So we will calculate this for V1. V1 is C2 V2 upon C1. How this formula is solved? For example, I want to calculate the V1 for 1.5. This is the concentration that I want to achieve. So I'll place 1.5 in the value of C1. I'll multiply it by in the value of C2, which I want to achieve, and the volume of V2 30. So this stays constant. 30 is always constant. So I multiply 1.5 by 30. I divide by the concentration 1, which is 2. So I'll get the answer 22.5. So I need to add 22.5 of sucrose solution. And to this sucrose solution, I need to add 7.5 of distilled water to make the final solution to be 30 cm3. Now, for the next, if I want to calculate for 1.0, what I'll do, I'll place this 1.0 here. This will be 1.0 multiplied by 30 divided by 2. I'll get the answer. The answer that I'll get, I'll place it here. And then subtracting it from 1, I'll place it for the water. Now, same I do for zero point 0.8 and zero point 0.5. You just fill up this table. This is something that you do in your P3 and AS as well. So describe a method the student could use to collect the data needed to determine the concentration of sucrose solution that has the same water potential as the cells of the beetroot. The student was supplied with sucrose concentration prepared by standard lab equipment. Do not include the details of how the student prepared the sucrose solution, which means dilution details are not required. Would use the data to collect, determine the concentration. That data is not required. Um, your method should be set out in a logical order and detailed enough for other person to use. So what have we done? We how many solutions do I have? I have 1, 2, 3, 4, 5, 6. I have these 6 different solutions here. So what I'll make, I'll do 1, 2, 3, 4, 5, 6. I'll take 6 beakers. In each beaker, I'll just place the appropriate concentration of that particular sucrose solution. And to that, each of them, I'll place the beetroot section as well, cut in an appropriate block to keep the surface area volume ratio constant throughout. And then I'll just wait. For example, the beetroot, same taken, it is cut with a scalpel into a block or strip, dimensions of surface area volume ratio 2, 2, 2, 2 by 2 by 2 by each. So I'll place like for instance, I'm taking 18 blocks whole. I need 3 repeats, so I'll use a scale for measurement. I'll measure the mass of each block initially. I weigh them on electronic balance, and then I immerse one block in each of the beaker, each like 2 mole per dm cube, 1.5 mole per dm cube. I make set of 3 beakers for each particular concentration with one block fully immersed. I leave the this apparatus for 30 minutes, and after 30 minutes, I'll just take out all the blocks from each of these and I'll reweigh them. When I reweigh them, I'll find out the difference in the mass because I might have absorbed water, or they might have let the water go out. So I'll find out the mass after 30 minutes. I'll just wipe it with the filter paper and wipe away the excess solution. So for each sucrose concentration, and then I'll mean change in mass will be calculated. I'll plot a graph of sucrose concentration on the x-axis and the change in the mass on y-axis. The scalpel, is a source of risk. There's a risk that you may cut your skin. So the scalpel is sharp, handled with care to prevent getting cut on the skin. So you mentioned the risk, the hazard, and the precaution. Okay, then they are saying how the student calculated the mean percentage match in the mass of the beetroot for each concentration of sucrose solution. The student plotted these calculated values to obtain a line graph. Complete figure 1.2, labeling the axis, sketching the lines that the student obtained. So we can expect the sucrose concentration in mole per dm cube and a change in mass in milligram to change in this way, that at 0 it should be maximum, and then as you start to increase the sucrose concentration, the change in mass decreases, and then a point comes where there is no change in mass, and this no change in mass is the point which is the same as the sucrose concentration is the same as that inside and outside. So that's the point of intersection that tells you the precise sucrose concentration present inside the beetroot. So this is what they are asking you. In the next part, you can just mention it that way. The cells of the beetroot contain a red pigment that can leak out in solution that is cut. The student noticed that each sucrose solution contained a colored red at the end of investigation. So this one improvements would make to method reduce the amount of red pigment of each. So what do? You can when you cut this beetroot, the red pigment comes out, the solution becomes reddish. So what do? You can just rinse it with running water before immersing it in the sucrose solution initially. Question 2 deals with a buff-tailed bumblebee. The name is mentioned, is in an insect that uses its tongue to feed on the nectar of pollen of flowers. Bumblebee can feed on nectar and the pollen of flowers by either gripping and holding, these are one way, or just onto the flower petal, or by hovering on the or flying in front of the flower. So these are two ways in which it can either feed on this. So this is the bumblebee shown, and the claw. This is the claw that it will use to hold, and this is the whole bumblebee. Next, they are showing to you how the bumblebee can help to grip the claws. It uses the claws to grip the flower it's feeding on. In the next question part, they are showing you a figure with the claw magnified, and the claws of the bee are shown. So suggest a method that can be used to measure the image length of the longest part of the curved claw shown in figure 2.3. So how can you measure the longest part? So you can just place a scale here and a scale here and just measure between the two claws, same part on the other claw. So you just describe it properly using a ruler, measure the width between the claws from inside curved part of one claw to the same part on the other claw. You can't measure it from here to here and say that this is the claw width. Similarly, you can't measure it from here and till here to say it's claw width. No, you have to measure from the same part at both claws to get a straight, you can say, distance between the two claws. Surface of petals of different plants have different textures. This show the electromicrograph of a rough surface of flower petal. So this is a rough texture. These are the bumps there. And this is a smoother surface of the flower petal where the bumps are not there. A scientist made an artificial flower apparatus, figure 2.6. There was a 60 millimeter diameter dish to represent the flower petal. A small drop of sucrose solution considered was representing nectar. The support was there. To model the flower petals of different levels of roughness, the scientists used different discs. So rough discs to and rough petals and smooth petals means rough discs and smooth discs in this case. Each disc was made using particles of different diameters attached to the surface of disc to make sure that they are of different roughness. Mean particle diameter will be your independent variable here, and this is the mean particle diameter. And as you increase the number of mean particle diameter, the level of roughness increases as well. So this is given to us. The diameter of each disc was measured as 60 millimeter. Identify one other variable the scientist should standardize when making the disc. So color of disc is very important because the insect will be attracted to an appropriate color. If you change the color of the disc, the insect may or may not hover. Similarly, the type of roughness of particle should be different, should not be different, should be made consistent. Each of these should be of the same texture. Okay, the scientists investigated if the level of roughness of disc affects whether the buff-tailed bumblebee grip or hover when they feed on the feeding visit. For each dish, the scientists recorded the number of feeding visits the buff-tail gripped it, or the number of like visiting for feeds and then hovering, or and then calculate the percentage of feed visiting from this that gripped onto the disc. So these three things were calculated. State the independent variable of this investigation. Now, whenever we are mentioning the independent variable of the investigation, independent variable is always the cause and the dependent variable is always the effect. So level of roughness of the mean particulate diameter is the independent variable or the cause here. For the disc with the mean particle diameter of 16 micrometer, the student calculated the buff-tailed bumblebee gripped at 79% of their physical feeding visits. The total number of feeding visits were 717. Calculate the number of feeding visits when the this bumblebee hovered in front of the disc. Give your answer to the nearest whole number. So how do you do? Total feeding visits are 117. Bees gripped it for 79%, and it hovered it for like how many? 79% mean 100 minus 79, which means 21%. Always make a data of the values given to you and then start to solve it. So things become much convenient. So 21% are the total visits which are there. So 21% of the total visits we have to find out. So 21 upon 100 multiplied by 117 gives you the answer, which will be rounded out to 25%. Okay. The results are shown. Mean particle diameter is increasing. When it's increasing, the feeding visits are of gripping changes. And eventually for 30 and 53, it is the same. There is no change there. State two conclusion that can be made. So increasing the particle diameter increases the feeding visits. Bumblebees gripped onto this. The increases as well. The 5 micrometer particle diameter bumblebees only hovered. So you can see that here, they are not gripping it at all. So when you increase the particle diameter, they start to grip it. Then you are asked to calculate the RS value. RS values are the rank, the Spearman rank correlation. There is no correlation between the mean particle diameter of the disk and the percentage feeding visits. So this is the null hypothesis of the student for using the Spearman rank. What you will do, you will solve this formula, n cube n d square. So where do you get all these values? So this is mean particle setting is 1, 2, 3, 4, 5, 6. So your n will be 6 here. The ranks are already given to you. What will you do? You will just rank the percentage of the bumblebee in an order. For instance, if this was ranked with the smallest value being 1, then the larger being 2, and then 3, and then 4, and then 5, and then 6. So again, we will rank these in the same order. So this will be 1, this will be 2, this will be 3, this will be 4. Then both are the same. So they will be 5 plus 6 divided by 2. They will give you 5.5 each. Difference of the rank is calculated and then it is squared. So you just calculate the difference between the ranks. So this will be 1 minus 1 gives you the value. 2 minus 4 gives you this. 3 minus 3 gives you this. And then you just square these things to get the d square value. Once the d square values are calculated, you sum up the d square values together. When the d square values are summed up, they come to 0.5. You just put the values in the formula. 0.5 for some d square multiplied by 6. Divide by 6 cube, multiply by 6. This 6 is part of your formula. So you get the sum d square value from the table by adding the things together. N cube and n are the number of samples that you took, which are 6 each. You calculate the whole thing in the bracket, then subtract the answer from 1. And the final answer comes out to be 0.997, rounded out to 0.99. This shows the critical values for at zero point 0.5 probability. So for 6n, it is 0.83. So we know our RS value is 0.99, which is greater than 0.83. So this means that the null hypothesis is rejected. It's a strong positive correlation exists between them. Question 3 deals with pectin, a polysaccharide found in the cell wall plants. Cell wall of plants. Pectinase is an enzyme that hydrolyzes pectin. Us to investigate the use of pectinase enzyme to extract juice from different fruits. So different type of fruits will be the substrate, and the enzyme is pectinase, which will act on the substrate pectin. So what did it do? They removed this particular skin, and they cut it into small pieces. They separated into beakers 5 cm cube each, and added the enzyme there, incubated for these minutes, and at this time, at this temperature for this time for hydrolysis to complete. And once they added into the funnel, so to collect the final juice of each type of fruit, volume of juice was calculated. This is given to you: type of fruit, sample 1, sample 2, sample 3, and mean samples are given. So the volume of the juice is calculated. You need to calculate the mean here. So you just add the three values, divide by 3, add the three values, divide by 3. So you calculate all these values here, and then you find out the mean simply. The independent variable is the type of fruit, which is the cause, and the effect is the volume, which is being extracted. So they are asking you the type of variable which is there. So the type of variable becomes like the type of fruit is the qualitative or a categoric variable, because apple, orange, these are different categories of fruits, and the volume of juice calculated or found out or taken out is a quantitative measurement. So this is the qualitative variable, this is the quantitative variable. What type of data are we getting? For type of fruit, it is a nominal data. Nominal data is something that you cannot arrange in order. And volume of juice extracted is a continuous reading. So that's why we are saying type of data continuous. State 2 changes in the student would make to their method to improve the quality of the result. They standardize the mass of the fruit of the piece, use boiled denatured enzyme for this. Every fruit should be of the same age. This will be standardizing our whole protocol. The last part, the student want to determine the quantity of pectin in the cell walls of different types of fruit. Suggest why the student could not use the experiment 3A to determine quantity of the pectin in the cell wall of different cell type. Fruit, different types of fruit. Why is that so? Because cell wall composition vary. Every fruit will have a different composition, so pectin will be different. It will not be same. So all fruits do not, number one, and another feature, they all do not ripen up at the same time. So they have different ages, and this will start to affect the quantity of the juice extracted as well. So these were the three questions in this paper 5. I hope this video was helpful for you. I'll end the recording here, and I hope this helped you to an extent. That's it from my side. Thank you so much.